All right. I think it's time we could start the event. What do you think, Amir?
Sure, Kshipra. Yeah.
Awesome. All right. Good morning, good afternoon, good evening to everyone joining in all over the globe. Appreciate that you have taken your time out to attend our event. It is April 29th here in the United States, and we are going to be discussing a subject of space biology here. I am Kshipra Kapoor, a final year PhD student at Rice University, Houston, Texas. And I'm going to be joined by my colleague Amir Nasiri from SNEV, as well as the distinguished Dr. Heath Mills, who is going to be talking a little bit about how Rhodium Scientific supports academic projects going to the space, his career journey, as well as his scientific journey, and some insights about space exploration and doing science experiments in microgravity. Before I begin the event, I would just like to go over a couple of rules of engagement before we move forward and then have an introduction from Amir, the president of SNEV, followed by the talk from Dr. Mills. So after the talk is over, we would open our platform for Q&A. And due to the nature of this talk and Zoom environment, I would request all the participants to have their questions in the chat box, and we would address them for you after the talk is over. The questions can also be anonymously asked. In order to do that, you could message me or Amir directly, and we would address those questions for you. You could also use the raise hand feature to add a comment or address something in the second half of the event. Also, feel free to use our media handle SNEV Research on Twitter to ask live questions, or you can also use Discord channel. And without any further ado, I wanna pass the mic over to Amir to do a quick introduction about SNEV, its history, and what we are up to, and then we would go into the talk by Dr. Mills.
Thank you, Kshipra, for your introduction. Hello, everyone. My name is Amir Nasiri. I'm final year PhD student at Latrobe University in Melbourne, Australia. And also I'm the president for a student network on extracellular vesicles. I just thought of take you briefly through the SNEV, since this is our first space biology talk. So SNEV is a student network on extracellular vesicles, and we are a team of international students where we officially started this network back in June 2020. And since then, we're running several events such as industrial talk, postdoc talk, and also virtual conference that's really focused on students researching on extracellular vesicles. But also we came up with this initiative where we want to start like a space biology talk. And today is the first space biology talk. And without waiting more, so I'm very excited about today's talk, and I just want to introduce our speaker today. So it is my great pleasure to introduce today and a speaker, Dr. Heath Mills. So Dr. Heath has over 18 years experience leading international research efforts to characterize and understand the life in extreme environment. He graduated from Duke University with Bachelor of Science in Biology and from the Georgia Institute of Technology with a PhD in Applied Biology. Dr. Mills has had an academic position at Florida State University, Texas A&M University, and the University of Houston Clear Lake, where he published nearly over 40 publications describing the extreme environment microbial ecosystem using advanced molecular techniques. Heath has championed extreme environment research by leading international collaboration, advising governmental agencies, organizing specific scientific conferences, and teaching collegiate level course in 15 different countries. Over the past 6 years, Mills has been a principal investigator and co-investigator on over 20 international space station science and engineering project funded privately and by the Department of Defense, National Science Foundation, National Institute of Health, and the International Space Station US National Laboratory. Recently, he joined Rhodium Scientific, where he served as the Chief Scientific Officer, leading initiative to expand terrestrial and space flight research capacities into the advanced commercial biotechnology sector. In space-based research, he has been co-principal investigator and principal investigator on multiple projects funded by the National Science Foundation, National Institute of Health, and the International Space Station National Laboratory. Currently, Heath is designing and managing Rhodium Scientific's portfolio of diverse, quality-controlled, enhanced biological projects for US and international research clients. Please help me to welcome Dr. Heath Mills for today, our first space biology talk. Thank you.
Thank you, Amir, and thank you, Kshipra. I really appreciate this today, and thank you for that great introduction there. I wish I'd recorded that. That was really cool. Thank you, Sarah, very much. So let me go ahead and share my screen here and start up my presentation for today. Here we go. So we're really interested to talk to everybody about the topic of space biology and some interactive ways that I've gotten involved in doing research and science in space over the years. I'm hoping to close out my talk in about 25, maybe 30 minutes to give us plenty of time for questions and answers at the end. So I'm really interested to hear your engagement points and where you are right now with your research, and hopefully possibly inspire you to start doing some research in space as well, and where your work can apply to this new field. And so, as Amir was saying, I'm going to go through a little bit of a background on this, but what I want to focus on this first slide right here is also who I'm working with and what I'm doing at Rhodium Scientific. So you'll see Rhodium Scientific's logo up in the top right corner. You also see in the bottom right corner my email address. At any point, make sure you write that down, and I'm accessible. Email me with questions, especially after all this is over with. But at Rhodium Scientific, I'm the Chief Scientific Officer. And I really hold two different positions within Rhodium. One of those is to help advance new biotech initiatives for the company. And in doing so, it's also really within the field of space biology, because this is a field that is evolving and it's changing, and as biology does, so does this field. It evolves and it changes. And so I'm always looking to develop new scientific missions and new technologies and exploring new research areas. But then I'm also working as the lead for the integration, the mission integration and operation side with NASA, which means I help move the projects through the safety integration, the onboarding process, and the eventual operations up on station. And that's what Rhodium Scientific really specializes in, is doing work, doing research, advancing research in space. And Rhodium Scientific is a woman-owned small business founded in 2014 by Olivia Holzhaus, who is our CEO and founder. We are an official ISS US National Lab implementation partner, and I'll get into more of what that means in some later slides. And we really try to focus on that full turnkey approach to getting your research into space and allowing you to do your research. We'll take care of the NASA stuff. We'll take care of the rocket science and let you do your bench science. And hopefully you'll be inspired by some of what we're going to say here and say today. So let's jump out way back into the beginning. And so one thing I was asked to do was kind of bring you guys up to speed on how I got here. And as most of y'all are right now in an academic position and going through graduate school, you're probably wondering why you are where you are and where you've been. Well, in my career, in my life, I really started as probably some of y'all have. We looked out a window and we stared up at the moon and the stars and wondered what was up there and wondered what could be going on in space. And so this is where it all started. And over a number of years, these years are getting longer by the day, this is where it's now going. This was us. This was the main core team at Rhodium Scientific. Our Dr. R.P. Oates on the left. There's Olivia Holzhaus in the middle, and I'm the guy over the goofy guy on the right. This is us enjoying some VIP access for SpaceX 20 launch back in March, just before all the COVID stuff happened. But we're sitting outside the VAB, the Vertical Assembly Building at Kennedy Space Center, celebrating a beautiful early morning launch there for Rhodium Scientific's mission. So how'd I get to there? What's the gap in between? And so, yeah, I was one of those space junkies as a kid. I'm an alumnus of the space camp in Huntsville, Alabama. I was there as a kid and got to go through all the simulators and got to do all that and play an astronaut a long time ago. I even taught a crazy little course back at Meredith College in Raleigh, North Carolina, to some 10 and 12-year-old students about space and building small miniature rockets. And yeah, I was 13 years old teaching some 10 and 12-year-olds. So it was a fun experience, but got me doing some science and got me doing some space very early on. Well, I carried that forward, and let's see. Let me see here. Hang on one second. There we go. So carried that forward off into my academic career and got into Duke University, where I really wanted to explore science. And so I didn't stick with one lab. I actually ventured around to multiple different labs to understand what science was all about. And in that, and learning different fields and different ways of conducting science, one thing really stuck out with me. And that's the summer that I got to spend down at the Duke Marine Lab doing some field work, doing some exploration, going out into the unknown. And that really got me excited about doing science. So I carried that excitement to my graduate work at Georgia Tech, where I stayed with biology, got into some molecular genetics, some environmental microbial ecology, but I also went out to sea. I did multiple oceanographic expeditions, 7 in all. I got 4 submersible dives down to the bottom of the ocean, with the deepest one being about 1.5 miles deep. Again, pushing the envelope of where biology exists and persists, and what that evolutionary pressure and stressors are of those extreme environments really intrigued me. So after graduate school, I took a postdoc position at Florida State, where now I moved a little bit outside of the biology realm. I got into a geochemical lab, a biogeochemical lab, and I started learning to be more interdisciplinary, starting blending languages, blending ideas, trying to understand more of a holistic view of how the environment works. I used that understanding and knowledge when I got to my academic positions at Texas A&M and University of Houston Clear Lake, to continue to, and that's not a typo here, if you can see my pointer, I do capitalize the B, the G, and the C here because I want it to be bio, geo, and chemistry. I want to really blend the 3 and not just use that as a catchphrase. And so using biogeochemistry, using molecular genetics to extend my understanding of how life exists in these extreme environments. And after academia, and after writing a lot of papers and a lot of grants and teaching a lot of classes, I decided to make the jump into the commercial space sector. It also helped that being here at the University of Houston Clear Lake, being right outside Johnson Space Center and talking to a lot of the NASA engineers and scientists, I started realizing that NASA was really where I wanted to be and working with the commercial space sector. So got into and started working with the new space field. I also then started learning a new language, and that's called business, where you don't get taught that a lot when you're going through academia, and especially in a science field. So I needed to learn new phrases and new terminology and new ways of literally doing business. And our CEO, Olivia Holzhaus, likes to use the hashtag business of science. And I'll tell you, I'm learning a lot more of that business of science. And again, we got those capital letters in there, just like my biogeochemistry background. So that's now where I am. That's now where I've gotten to. And where I'm sitting today is now the Chief Scientific Officer for Rhodium Scientific. But now what my job is here is to really stimulate that new market, that new sector, that new biotechnology sector. And so I like to really go into the why and the how. Why go to space? As a biologist, why go to space? As a biologist, how do you get to space? How do you get your research to space? And so that's the 2 areas that I want to focus on now. Let's see what happens here. So the commercial space industry right now is a very interesting and evolving field. NASA has done their job. NASA is doing their job. NASA is advancing their job out further to the moon and to Mars. Well, what that's doing is it's leaving space, and pun intended on that, leaving space behind for the commercial industry to fill in and to start taking advantage of those technologies and that research that NASA had provided us for over the many, many decades of going into and living and persisting in space. And so what we now get to operate on and what we get to focus our efforts on is currently the International Space Station. And yes, at certain time points during the day and the evening, you can see the International Space Station as it glides overhead. There's some good apps for you guys to check that out. But the International Space Station is and maintains a national lab. There is a U.S. national lab on the International Space Station. With me, with an oceanographic background, I also like to think of it as a field station. This is a research station to go into a novel environment and to explore and understand that environment. So it's a national lab and it's a field station to do your research and development. Now, what does that look like? Now, the International Space Station is about the size, and if you put it into regular terms, it's about the size of a 5-bedroom home. It stretches out end to end about the size of an American football field, so a little bit more than 120 yards in length, less than meters. But it has these different modules, and the module that we're most interested in is this guy right here, Destiny. And it's this Destiny module that's right here that houses the U.S. National Lab. That's where most of the equipment, most of the hardware, most of the research capabilities are that we access. Now, we can also operate within Columbus, which is where the European Space Agency operates and does a lot of their work, and also in the Japanese experiment module over here. But for the most of the work that we do is inside Destiny. And this is a lab, and it does say on the top that international collaborations are welcome. We do international work, and I'm going to talk about an international program at the end. So just because you're sitting in another country right now doesn't mean you get to go pay attention to something else. Your research can be and can focus through this work and through what I'm saying today. So please understand this is through international collaborations. So as a microbial ecologist, as an environmental microbiologist, as a biologist, I always want to look at these new environments and try and understand it from a biological perspective, from an environmental perspective, because as we all know, an environmental stress equals a biological response. When there is a stress on a biological organism, the biological organism responds in a new way. Now, typically what we look at when we culture, whether they're stem cells or fibroblast cells or microbial cells or fungal cells, take your pick, you have to pay attention to your media's salinity, the oxygen, the temperature, the nutrients, and it's dinner time here, I'm hungry, it's a cookie, but the nutrients that you provide your cells, your samples, and the overall radiation, whether that's light or it's high energy radiation, but different aspects of that environment produces the biological responses that we all study. Then the question becomes, is microgravity an environmental stress? And the overwhelming answer to that, and the overwhelming answer to the research that's been done by different space agencies around the world is biology changes in space. Thus, microgravity must be a stressor on biological systems. Understanding that stressor, now you get to harness this environment. And so what I want to do now is talk about some of those biological changes that have been observed over the years. And hopefully some of these will resonate with your current research objectives and current research interest. And so very simply, cellular growth, cellular morphology, cellular development is impacted by space. I have a handful of articles here, journal articles here, and as any good academic would do, I go back and I do a lit search and I try and understand that field. Picking out a random assortment of different papers here, you can see that cellular biology changes, the metabolic rate changes in space. There's alterations in that secondary metabolite production, what the cells produce. These growth rates, the cell densities change. Many of the fundamental aspects of how cells grow are altered by the microgravity space environment. One area of a particular interest to a handful of you out there I know right now is with cancer cells. Cancer biology is a very good thing to study in space. There's a lot of good research out there. Go do some lit search. I got some things on here right now about some cancer biology. Cell growth, morphology, development changes in space. Now, another fun one is virulence, pathogenicity, biofilm formation, some biofilm formation associated with pathogenicity, resistance to antibiotics, to immune systems. All of these are impacted by space. There's a handful of papers here that show that and prove that. Some of these are review articles that then lead to many other papers. Surface adherence, aggregation of cells. You don't have that settling effect in space that you have on Earth. So what proteins are now expressed for surface adherence? Pathogenicity of the cell changes. Some pathogens get more pathogenic. Some get less pathogenic in space. A lot of these changes appear to be epigenomic, meaning that they are shifts in the transcriptome. They're temporary shifts in the cellular function, a response to that environment. Why? What are those triggers? What causes them to be resistant to antibiotics? What causes the human immune system to change in space? These are all fundamental questions that need to be explained and need to be explored further. Go back to the molecular level, gene expression, DNA repair, even out to the full metabolome itself gets altered in space. Now, some of these, again, the transcriptome side, the proteome shifts, these changes in microgravity are at this molecular level. Now, this applies to, say, biomanufacturing. If you're up there trying to produce a drug compound to support exploration, if you're trying to produce a novel drug compound to bring it back to Earth, you have to go up and understand these rate changes. Even down to repair mechanisms, and this could link back to cancer, it could link to aging-related diseases. Some of these, due to the low persistent dose radiation that happens in space, make studying radiobiology in space a very intriguing field. Now, I'm sure a handful of y'all were really hoping I'd have a slide on this, since it is the group we're talking to today. And yes, extracellular vesicles do change in space as well. What research has shown, and I will say, and if you look at the fourth bullet point on here, more work needs to be done. But from the limited studies that have been done in microgravity, it is showing that the quantity and morphology of extracellular vesicles change in space. Even the content of these extracellular vesicles change in space. Now, could that be because the cell is responding to this stress and is then exporting different compounds into these extracellular vesicles? Most likely. Is this a stress response to the cell? Yes. But if you're using these extracellular vesicles as that diagnostic tool for either some type of disease, some type of stress, this is a diagnostic that could be developed for advancing the field and going into space and persisting in space, but then also understanding, especially some of like cancer again and aging-related maladies. So yes, space impacts extracellular vesicles as well. So the group is on the right track on that. So now, one thing I had to do as I was going from the academic approach of going and doing these lit searches and identifying the research and identifying the gaps in the science field that I was in, is that as I transitioned into the private industry and the commercial space industry, what then I needed to look at was, okay, we've discovered this, now how can it be used? That discovery to applied research is now where I really focus in. How can those findings be applied to drug discovery and drug development? Regenerative medicine with tissue and organ production. As I mentioned before, biomanufacturing. And also in synthetic biology as well is becoming a very hot topic in space. And so now what I'm looking at and what I would love to talk to you guys about today is how to advance some of your research that you're doing into an applied nature and utilize that microgravity environment. So that's the why. And so I'm alluding to the how. Let's get into that how now. Let's go into how you are able to conduct your research in space. Now, we would hope that, and as I'm sitting with a nice logo on my shirt right now, we hope that you use and join us at Rhodium, and we can help you guys get your research to space and bring it back. We are, and as I mentioned before, we do have multiple access points to space. We are an U.S. National Lab implementation partner. We are also partnering with other companies to provide access to launch vehicles and new and developing orbital platforms. We also provide, as I said before, part of my job is to help support the mission development, from designing the project, helping you walk through the process of getting your project translated from what you're doing on the bench to what's able to be done on the space station, manage that project and do the full mission integration and operations side. Now, one thing that Olivia really puts forward with this company is that we maintain a high level of quality assurance, because this is, again, is a commercial company. We're trying to produce commercial products. And so we do follow that quality assurance protocols. Now, What Rhodium has been able to do over the past several years is develop some flight hardware that you would be able to use, and also some other flight hardware that we're able to bring in from other companies. But Rhodium has developed an ISS facility, the Rhodium Science Chambers. These are able to support cell culture rather, and microbial growth cultures. We also have the Rhodium Science Temp Log, a very unique, amazing device that we partnered up with iButton on to produce. This device flew and supported multiple missions last year. And I'll show you a picture of it coming up, where it's able to monitor and measure both temperature and humidity. And as I said, we can also identify new hardware that may fit your research needs. And so we are, and here's another one of our old pictures here, it keeps popping up. We are a science company. And so whenever we work with you, we want to develop that science first. We want to make sure that your ideas are captured in these flight protocols. And so we do focus on that science strategy first. We use an old tactic that NASA did back in the early days and still does today. When talking to the astronauts on orbit, you have an astronaut act as that go-between. They are able to speak the language and understand what the astronaut on orbit's going through. We follow that same mantra where we have scientists talking to scientists to help that project translation going forward. So we'll help with that. We'll walk you through that. We'll provide that opportunity to get your science understood. And then we've developed this new process, the quality industry compatible space process, the quick space process. And that's where we really interject that quality assurance into every mission. And speaking of missions, we had some fun last year with some patch designs. You can see down here, we flew 5 different missions last year. Phage evolution, space microbiome, in-flight manufacturing, biomanufacturing, the temp log, and space rhizosphere. And I'll talk about some of these coming up and show you exactly what I'm talking about, what we're able to do with this process. And this is your access point. This is where, if you are interested, if I've gotten you at all inspired in this, this is the process that goes forward and allows you to get to space. And so what Rhodium has established is the Rhodium International Space Science Program. And we're right now using this program. I was talking to Amir earlier about doing this with Swinburne University down in Melbourne. We're doing this with multiple different projects worldwide. What we do is we try and get you onboarded to the International Space Station and back and finish out your experiment within about a year. And that allows you to either operate on business timescales or academic timescales. Now, this is a program. It can also be for individual scientists. If you have an individual project idea, we can ramp you into this. If your school, your university, your industry, your company wants to get involved, we can put a company or a school into this, or government systems, working with multiple governments to be able to put this in play. So this is a process where we'll walk you through and get your science onto that International Space Station and do the research you're wanting to do. So what's been done? So I showed you all of those experiments before, and those data and the research before, and those other publications. Here's a glimpse at a couple projects that we're right now finishing up with Rhodium and with some of these partner universities and entities. One of these is phage evolution. The top left corner is the operational nomenclature for the project, NASA's OPNOM, as they call it. It is searchable as phage evolution. And what this was looking for is whether or not a bacterial phage is a virus that only attacks bacteria. Whether or not this bacterial phage and their host, the bacteria that's susceptible to that phage, how they evolve in space within this new environment with new stressors. How do they change, and what changes that interaction between that predator-prey system of that phage and that host? Well, this flew last year to the International Space Station aboard Northrop Grumman 13. It flew last right before COVID hit. Data sets coming out now show that we have some interesting findings, and we'll be publishing on this over the summer, that yes, the phage evolves, it changes. The way it interacts with the cell changes in space. So even phages and that virus changes and responds to that environmental stressor. At Clemson University, we worked with a project there, also funded by, and that previous work was funded by the Defense Threat Reduction Agency and supported by the ISS National Lab. So is this project here. Oh, and again, that's us over on the right side here. I forgot to say this. This was us celebrating launch with the lab and with the lab from Wisconsin, Dr. Batson Rahman's lab at the launch site. Here is Olivia and RP doing the final processing for the payload for rhodium in-flight biomanufacturing. This project was looking at that biomanufacturing aspect of microgravity research. So if you know the production rates on Earth, are those same production rates seen in space? Is it stable? Does it change? Is it predictable? What are those rates when gravity is no longer there? And so this is very important for the Department of Defense for point source manufacturing. And so right now, this project is being analyzed. It came back to Earth last summer, and we're right now looking forward to seeing some good results with this one. And so we'll have to come back and fill you in more on that one. This one here was a project with Los Alamos National Lab. This is rhodium space microbiome. The microbiome in this case was the gut microbiome. And as astronauts go to space and space travelers go and leave Earth's gravity, they experience not just the human experiences microgravity, but all the bacteria within the human system, especially the gut microbiome. A lot of research now shows and points to a healthy gut microbiome as now having a healthy host, a healthy human. As you change that gut microbiome composition, you also change the overall health of the human. So what happens in space? What happens with that gut microbiome as it changes? Well, the data sets that are being produced right now and the preliminary data suggest that yes, the microbiome changes. Yes, there are hidden pathogens that now show up more prevalent than before. Some beneficial bacteria lose abundance in space. And so you take that information, you start blending it together where now you start looking for countermeasures. How do you protect human health? By understanding those shifts in your gut microbiome. So that's just some of the projects that we've done. That's some of the projects that we're helping support. And now we get to the part that I've been most interested in. And that is you guys and your questions and your interest and where your research is wanting to go. I'm going to open up the floor to some questions. I want to very much thank both of my hosts, Amir and Kshipra. Thank you very much for the invitation on this. Thank you to the Student Network on EVs to get me here and have me do this, and especially the NASA JSC Speaker Series for connecting us on this. And so let's take that first step. Let's see where your research is able to go, and let's start that journey. So thank you, everyone. Thanks, everybody, for letting me do this. And I'm going to stop sharing for now and open this up to some questions.
Thank you so much, Heath. It was a fantastic and very informative talk. We already have questions coming up in the chat window. The first question is, do you cook the microgravity field for cell culturing on the Earth?
Okay, so I'm assuming what you're meaning is that do we have simulated microgravity environments on Earth, and can you utilize those on Earth? And so the answer is there are simulated microgravity machines on Earth. There's a rotating wall vessel. There are random positioning machines. The answer on that, or the caveat on that, is depending on the cell system you're working on, sometimes these systems work well. And sometimes these systems actually produce a third phenotype. And so you have a terrestrial phenotype, you have a space phenotype, and then you have a simulated gravity phenotype. And so there is some testing that needs to be done to validate some of these systems with your organism or your cell system or your question. But there are some simulated microgravity devices on Earth. If you are really intending to go to space and support the astronauts and support exploration or support development of work in space, our suggestion is go test in the environment that you're going to be in. You can simulate seawater in your lab, but you really have to go to the ocean to understand oceanography. So that's the basic answer on that.
Thank you, Heath. We have a second question, and it says, with the limited ISS on orbit hardware, how is Rhodium working around with these hardware limitations?
Thanks, Mark, for that question. I know Mark really well. And Mark, you are exactly right. There are limited on-orbit hardware capabilities. One thing that we really try and do at Rhodium and what I've done through my career doing a lot of oceanographic work in some crazy locations is that constrain your experiment to space. Constrain the most important part of your experiment to space. And a lot with biology is that growth sequence, that incubation period. Have your cells acclimate to that environment, or your organism acclimate to that environment, grow within that environment, change because of that environment. And in many ways, the preservation of those samples becomes then key to bring it back to your lab back on Earth to do your final analytics. Now, that creates a bit of a log jam within the space industry, and bringing back a lot of samples and bringing back hardware is difficult to do sometimes. And so there is the need to develop new hardware capabilities, new onboard analytics. And so what we're trying to do is to test those new analytics that are coming online, those new experiments online, and help develop them so that we can do more research in space. The key to that is you really have to make sure that the hardware is miniaturized, that it's smaller, that it's ruggedized for space. And so we are willing to work with different groups to try and test, develop, and increase that overall capacity.
So the next question we have is, how do you see ground-based low gravity biological studies? Also, it's expensive to conduct on-flight missions. So it's, I think, pertaining towards cost of the mission.
Yeah, two very good questions. And I talked a little bit about the ground-based studies, and those are good to do. Those are good proof of concept to show that you do have a system that is changing in microgravity. Depending on your overall goal and your end target with your experiment, sometimes doing the simulated ground studies is sufficient and you can get the data sets that you need, or sometimes you have to go to space. One thing that is helping right now on the second half of the question, that expense, is that with more projects going to space, more science going to space, with more initiatives going to space, more funding is opening up and there's more funding opportunities. The overall cost is coming down. One thing that we try to do, because our hardware that we've developed is relatively small, it is hardware that is very simple hardware. It is hardware that you've probably worked with in your lab right now. We try and fly what you are used to in the basic scientific equipment that you grow your cells in right now. That's what we fly to space. So we reduce the overall engineering cost. We reduce that engineering price tag on these flights. And so then we work it down so that it is, and the flights are much, much, much cheaper than they have been just a few years ago. They fit within, in the US structure, within National Science Foundation and the National Institutes of Health, within their regular budgeting or funding cycles, with their regular proposals, you can now put in a spaceflight component to those. So what was cost prohibitive years ago, that cost a lot of money to go to space, it's now getting to the point where you can go easier to space and with a cost lower than some of the oceanographic trips that I did and expeditions that I did years ago. So I beg you to explore this cost on this. And one thing we do at Rhodium is A, we try and lower the cost, but B, we also try and help you find the funding. We write proposals, we write the grants with you. We go and we help identify new funding mechanisms with it. So again, we're there with you, we're part of the team.
That was very insightful, Dr. Mills. Next question is there any specific funding opportunities for these space-based research?
It was a great segue question. I'm glad you guys wrote those back to back. That was perfect. Thank you. Yes, there are dedicated funding opportunities for space research. A lot of those are through the ISS U.S. National Lab. They're also known as CASIS, or the Center for the Advancement of Science in Space. There are a lot of calls for proposals with that organization. And once when we understand what your research is, we help, hey, there's a call coming out, there's proposals coming out, let's write to these. NASA and space biology with NASA also offers many opportunities to go to space. But also NSF, NIH, other CSIRO out of Australia, other groups do fund space-based research. And one thing that changes the mind of a lot of program managers, I mean, also we're funded through the Defense Threat Reduction Agency, a division of the Department of Defense. What changes a lot of program managers' mind is when you understand and start looking at space as an environment. If you look at it as an environment with which biology changes, then it's no different than NSF funding an oceanographic work or NIH funding a project looking at sewer composition. You don't have to specifically be associated with the human body to do NIH work, or you can do ocean work with NSF. You can do space work with those two groups. NIH has funded space work. NSF has funded space work. Other program managers, when they see that this is in an environment to understand your system, extracellular vesicles, now you can apply to an average call, a normal call, a call that may not be specifically listing space as an option, but you're applying that environment to the question the program managers are asking. So that's something we can also help you with. We can help you with the language. We can help you with the documentation on that, because there are other funding opportunities out there than just the calls that may come out through a space agency.
Thanks, Dr. Mills. The next question is regarding imaging methods. So is there any way, or do you have any hardware methods for imaging?
Absolutely. So there are multiple microscopes currently on station right now on the International Space Station that can be used. There are small microscopes. There are some larger confocal microscopes that are available. There's also plate readers for 96 well plates that you can do fluorescence, autofluorescence on. Cell densities, looking at cell culture density, spectrophotometry on it. So there are imaging systems. There's video cameras where you can look at changes in morphology over time. So yes, there are dedicated imagery systems. There's always a need to advance those, and Mark will agree with me on this, that there's need to advance some of those imagery systems. But there are currently ways to do from microscopy to visual to different spectrophotometer fields. So yeah, there are ways to do imaging.
Thank you for those insights. The next question is actually based on one of the slides which you had pulled up with respect to the extracellular vesicles, which says, would it be possible to collect biofluid samples from the astronauts before, during, and after space flight from, say, plasma, saliva, or urine?
Absolutely. So this goes into the request for samples, and this goes into, there's actually an archive of astronaut fluids that is maintained. And so there's an archive for those. You can also collect those on astronauts that fly currently or that are on station currently. You can get before, during, and after samples to do many of the studies that you're talking about. And yes, one of the studies that I posted there with the extracellular vesicles was with some saliva work that one of the groups were doing. So yes, you can do that. It just takes some paperwork. We'll obviously navigate that for you, but that is something that can be taken and preserved if your sample requires, say, centrifugation, if it requires special temperature preservation. All of that can be worked out so the onboard samples can be processed and handled the way that you need to do your downstream analytics of choice.
That's awesome. The next question is based on animal models. What are the animal models that are currently being used at Rhodium, by Rhodium in ISS?
Besides RP and I think we're part of the animal models that Rhodium uses a lot. But we do support the rodent research mission. So we help provide some of the science validation and testing for some of the rodent research missions. So there are mice that go to station through different, both the Japanese Space Agency and through NASA. There are other fly missions, Drosophila missions that have gone up. I know some of the researchers that lead those Drosophila teams, and we've worked with them in the past. Obviously, microbial, fungal samples, tissue cultures. But for the animal models, the main ones are mice, Drosophila. There have been some zebrafish that have gone. But if you have other organisms, I think there was a student study at one point that actually did spiders and looking at spider web design in space, which I thought was awesome. But if you have an idea for an animal model, ask me, and we can identify how to do it or if it is possible to get that up there.
Rachel says, thanks for the great talk. And she also wanted to know what kind of research is being done at ISS, and have you identified anything that makes something a good project to take to space? For example, a project that has implications for astronaut health, or previous evidence of something that changes with environmental stress.
First off, thank you very much, Rachel. I appreciate that, the compliment there. Thank you very much. And you would be surprised about and I'm surprised every day about the breadth of research that's being done on the International Space Station, and it has been done in space overall, even back through the shuttle days. What typically makes a good space project is what makes a good terrestrial project. Know your constraints, know your variables, do your replicates, do your controls. And that's one thing that during the project development phase that I had up on that one slide that we really want you to focus on and understand. We want the research to be valid through all the reviewers in the peer review system. So make sure you have your controls. Make sure you have those replicates. Make sure you have enough time points. If you're doing a time point analysis, make sure you have more than just 3 time points. Make sure you spread out some time points on there. Make sure you have enough volume of your samples so that you can perform all the downstream analytics. And even if there needs to be some protocol shifts and troubleshooting, you have enough volume to get those results that you were looking for off the start. So what makes a good project in space is what makes a good project on your bench. And one thing that we try to do is that whatever vessel you use on Earth, if you're using a flask, if you're using a petri dish, if you're using a certain container size, volume, shape, trying to match that on orbit so that you don't have container effects that does plague some tissue culture down to microbiology. So different cells change just by the shape and size of the vessel it's in. So really pay attention to those details. That's what makes a good space project. And then whether or not you look at astronaut health or those that go to space, their health, or you are looking at the 8 billion people back on Earth health, that's both sides of those questions are valid. In the US with NASA tends to focus on health in space and help with the space traveler. The Center for Advancement of Science in Space, or the US ISS National Lab, tends to focus on the commercial aspect and helping those back on Earth. And so depending on which direction you go with your research is where your question lies. So both sides need to take advantage of that research. Again, going back to oceanography, we don't just do oceanographic research to help the fish out at sea. We do oceanographic research to help everybody back on dry land. So when you're thinking about doing space biology, you think about doing research up there in space to also help everybody else back here on Earth. And so both of those questions tend to be very valid. And then with those changes in environmental stress, that's the fun part. And going back through some of those papers that I posted up real quick, and if you do your own lit search, really identifying how they did their experiment, what was that stress, and then where do you fill in those gaps and advance that research to see what that stressor is like. There's a lot of research that can be done there.
Very interesting. So there's a follow-up question with respect to the microgravity. And it says, if this factor could reduce the cancer cell growth, how should it be applied?
Okay.
What are your thoughts on it?
Perfect. So part of, you don't want to have the therapy for a cancer patient to have to fly them to space to be cured, although I would gladly sign up for that trip. What you want to identify, and with the research that you do in space, and especially with a lot of the cell culture work and with the biological side of going to space, is whether or not it's a mutational change, which means now that change that happened in space, that stress has changed that organism at a genomic level, or it's an epigenomic response, it's a transient response to that environment. It's a temporary shift in how that cell behaves. What you want to do with cancer research and some of the stem cell, the regenerative medicine, those type of research is identify, is this a transient change, which means does the cancer cell turn off? Does the pathogen turn on? Does the cell get more virulent? Does the cancer become more aggressive in space, but reverts back to a wild type or a terrestrial phenotype when it comes back? If that's the case, then you're dealing with a transcriptome shift. You're dealing with gene expression. You're dealing with an operon. You're dealing with a repressor or an activator. Identifying what those activators, those repressors, those certain gene combinations are that elicit that response, now you're talking a therapeutic. Now you're talking drug discovery, drug development. So going up to space, constraining that research, that microgravity environment, taking that snapshot of that transcriptome, that proteome, that metabolome, those extracellular vesicles, maybe it's a cell signaling system between the extracellular vesicles that the cell gets stressed out and starts putting in a microRNA within the extracellular vesicle that tells the cells around it to respond differently, but it only does that when it's in space. Maybe you can induce that response back on Earth. Using some genetic engineering, some CRISPR technology, now you're starting to talk new therapeutics. So that's the type of research that I would love to work with you guys on. I would love to get involved in, and that's where the whole field is going. So you're right on track with that.
Very interesting point. So Nima Khalili mentions, thanks, Dr. Mills, for your fantastic talk. And as you mentioned earlier, you have worked on deep oceanic lives, bringing deep sea creatures to the surface. Can you put so many put so many stressors on them? What about moving far, far away and take the deep sea microorganisms to the space?
That's a great point. Now you're moving one organism from one extreme environment and you're putting it to the other extreme environment. And a lot of what's been done, especially with the deep subsurface work and the deep oceanographic work, is learning how a biological system adapts to those stressors, and then where those adaptations may be applied to other environments. And so when you think of a microgravity environment where certain physical stressors are no longer present on the cell, say deformation due to sedimentation in a vessel, certain pressures within the cell wall may be relaxed and changed. Well, in a deep subsurface environment, a deep oceanographic environment, those pressures and stressors are almost the exact opposite. They're being constrained and pressed in on. And so we know that there are shifts in the cellular composition, the proteins and the lipid structure within a cell wall under high pressure environments, in a barophilic environment. So doing that counter study in space, how does the lipid composition, the protein composition, whether or not they're more hydrophilic or hydrophobic, what changes within that molecular structure of those proteins and lipids to make that cell wall more adapted to that microgravity, low gravity environment? That's something that needs to be done. So maybe taking some of those organisms from this environment, putting them into space, maybe taking them from some of these dynamic environments on Earth. One of the most dynamic environments on Earth is a coastal system that goes from wet to dry, oxic to anoxic, sunlit to dark, pH ranges. These environments, these organisms are used to being in very drastically changed environments. Maybe some of those are more adapted to go to space. Maybe those can be some of the scaffold organisms that helps with biomanufacturing, with drug development, with new bioprospecting. We have a whole program at Rhodium right now for bioprospecting, looking for these novel organisms that do novel things that solve real world problems. We have a whole program for that here. And so you're exactly right. Finding those organisms and applying them to this environment is something that needs to be done.
Awesome. We are perfect on time. We had perfect Q&A session. And again, thank you so much, Dr. Mills, for giving such an inspiring and motivating talk to us. And I'm sure definitely an audience, someone or the other would have this idea of emailing you and taking it further to try and see if their PhD projects can land up in space.
Please do. Please email me. And the Rhodium website has more information on it, rhodiumscientific.com. We're on all kinds of social media, Twitter, LinkedIn, Facebook. So please follow us. Please email me with questions, comments, and let's go to space. Let's get your project up there, and let's get you a mission patch also about your project, and let's go to space. So thank you, everyone. Thanks again, Kshipra. Thanks, Amir. Really appreciate it.
Thank you. Dr. Mills, we would just like you to be on call for a minute.
Yes, absolutely. I'll stick around.
Yeah, thank you.
Thanks, everybody.
Oh, Kshipra, are you recording?
Yes, I recorded the event.
Okay. Do you want to stop it so you have